Hello, my name is Christian Rolfo. I am the head of Phase 1 unit in Antwerp University Hospital in Belgium and professor in oncology. And we will discuss today the liquid biopsies in lung cancer. My focus of research is lung cancer, and so I will orient all this presentation to lung cancer. That's are my disclosures. So the concept of a non-invasive tool like a liquid biopsy and how we are able to catch different components of the liquid biopsy family in the cells is easy to understand when we are thinking about the metastatization process. So the cells and the components of the liquid biopsies are traveling in the bloodstream and arriving to the new location to colonize these sites. So the idea to fish in these alterations in the bloodstream is very attractive. But before to speak about that, I would like to remind one of the important concepts that we need to be aware when we are working with liquid biopsies. The first one is the tumor heterogeneity. So if you see the case of the lung cancer, the group of population of lung cancer that we treat in the beginning with one only drug for all the comers, so with only combination chemotherapy for all the comers, when we start to see different alterations in the molecular approach, we have an stratification of rare disease. So you have patients with EGFR mutations that are 15% of the Caucasian population, 14% in Asian populations, but you have other drug-able targets like BRAF mutations, that is 1.4% in BRAF in lung cancer, but the activity of the drugs in these specific scenarios are very high. For example, BRAF mutations have an opportunity to have a more than 60% of response rate when you use the combination of the dabrafenib in combination with trametinib. So what happened with these patients after they have a recurrence? One of the mechanisms of resistance can arise like T17IM in the case of EGFR mutation, but also we need to be aware that some other mechanisms are also there, could be there, and we need to discover which one of these mechanisms is in charge of the progression in order to treat our patients in the proper way. So another concept that we need to understand is the temporal heterogeneity. So the tumors evolve over the time and develop mechanisms of resistance. So some sensitive clones that respond in the beginning are overcome by resistant clones that lead in the progression. And another important issue that we need to be aware is that the multiple steps require large tissue volume. And that is something that every day our pathologists are facing. We are, like oncologists, asking more and more determination because we have trial, we have new drugs coming, we have compassionate use, but the material is not enough to determine all the alteration. So liquid biopsies, and I'm referring like liquid biopsies, not only free DNA, but also CTCs, exosomes, microRNAs, long non-coding platelets that are traveling in the bloodstream are very useful for the determination of different scenarios. Important to remember that there are some concepts that, for example, when we are facing free DNA, not all the patients and not all the tumor types are releasing the same quantity or amount of free DNA. So we have also tumors that are shedding and not shedding free DNA in the blood. And that is very important to understand. We also need to consider that a majority of number of metastatic site, we will have more opportunity to have a major concentration of free DNA. And also, we need to consider the time point that we are catching for, we are using for catch the free DNA. If you have a patient that is responding to the treatment or have an oligo progression, it's very difficult that you will be able to catch in the blood free DNA for determination. That is completely different when the patient is progressing or when the patient have an important number of bulky disease. In the case of T17IM, that is one of the leading mechanisms of resistance of EGFR, you see that there is a high concordance between the plasma and the tumor, and the T17IM positive patients and negative patients were compared in this study published by Oxnard, and in order to the overall response rate are very similar. So we can say that the tissue and the liquid biopsies, and in this case plasma, determination are high concordance. FDA approved recently the use of osimertinib based in the presence of T17IM in plasma. So that's a very good achievement for our patients, that we don't need an invasive test in order to determine or to get a new drug. There are several tests that are studying, or several studies that are trying to determine the sensitivity in plasma compared with the tissue. The first line that you have there is the cobas in tissue and in the cobas in free DNA. cobas version 2 is the only one test approved for free DNA. It's still a very weak assay for the determination of T17IM. As you can see here, compared with digital droplet PCR or beaming, the sensitivity of the cobas-2 is very low, near to 59%. And so we need more assays that are able to determine that. We believe that next generation sequencing is the future for that. Actually, in our institution, we are using next generation sequencing and digital Doppler PCR as well. Obviously, tissue is still a major issue. So in case that you have, for example, in the strategy 1, you have a T17IM positive, and there is no problem that the patient will receive in plasma for plasma positive third generation TKI. But in case that T17IM is negative, you need to repeat the tissue biopsy, because the benefit that the patient can have with osimertinib is very high compared with chemotherapy. So tissue is still mandatory in this scenario. There are new unmet medical needs that are, for example, the new mutations, acquired resistant mutation for third generation TKI, like 797S. And in this case, we just published on our experience with a case report of this situation, 797 mechanism of resistance in lung cancer, specifically detected in liquid biopsies. In immunotherapy, that's another important topic. We come from a background of T17IM and EGFR in liquid biopsy, but here we are speaking about a complex microenvironment. So we have different opportunities. Liquid biopsy in immunotherapy is also an important topic. Because we come from an important background that is EGFR, T17IM, so a target therapy, a target that is able to be determined in that is able to be determined in the liquid biopsies. But here we have a complex microenvironment with several players. So having access to determine quickly PD-L1 from the blood is not easy. We have some studies performed in circulating tumor cells to analyze the expression of PD-L1 in their surface of the cells, but unfortunately the concordance with the tissue is very weak. Obviously, it's very naive to believe that PD-L1 is the answer of all the questions. So we need more tools to determine the prognostic and the predictive value of this biomarker for our patients. So one of the tools is the mutational tumor burden in blood. We know that the mutational tumor burden in tissue was analyzed in the trial of nivolumab and atezolizumab, demonstrated that it is a predictive biomarker for immunocheckpoints inhibitors. In the case of the blood, the study presented by Gandara in ESMO 2017 with a cutoff of 16 copies of mutations in the blood demonstrate that is a predictive biomarker for progression-free survival. Unfortunately, that's a great news, but unfortunately, it's not widely applicable in clinical practice. We need validation, but it's a very important new approach, and there are several research work coming to answer this question. In our institution, we also are working with exosomes. That is another component of the liquid biopsy family. We presented this data about ALK translocation, we call exo-ALK, and that was a proof of concept of patients to determine for patients to harboring who harboring ALK translocation. Sensitivity was 64%, specificity 100%. We are working in the validation of this data. So in the take-home message, liquid biopsies are entering the clinical practice, important tool for non-small cell lung cancer, but also in other tumors. Free DNA have a high concordance with tissue, specifically in lung cancer. Exosomes are the future for in the family. Also, we are working with platelets, and we are working with other fluids like saliva, urine, and pleural fluids. We need, obviously, cooperative groups, pharma efforts, and we just finalize, and we hope to publish soon, the ILCC consensus statement for liquid biopsies in collaboration with the panel of experts of ILCC. In addition, I suggest to you to be members of the International Society of Liquid Biopsy. That is a forum of experts who are aiming to have an organization for homogenization of and standardization of the liquid biopsy methods and education. This is the people who is working with me in this project, and I would like to thank you for your participation today. Thank you.